Starting from chapter number 4, that is your biological molecules. Okay. So in your biological molecules, you have your carbohydrates, fats, proteins. Okay. And you'll also talk about your DNA. So if we first talk about your, what exactly are humans made up of, right? So most abundant compound in our bodies is water. Okay, that makes up around 62% of our body. All right. So 62% of our body is made up of water. Okay. Then 16% is protein, 16% is fat, 1% around 1% is carbohydrate, remaining 6% are calcium and phosphate, that is mostly in bones, and some other elements as well, like potassium, sodium, phosphate, chlorides, all of these. DNA also makes up less than 1%. So main things that we talked about. Water makes up 62%, proteins make up 16%, fats make up 16%, and carbohydrates make up around 1%. Okay. First biological molecule that we'll be talking about in detail is a carbohydrate. So general elements of your carbohydrates, they are made from elements of carbon, hydrogen, and oxygen. All right. Okay. So one molecule of carbohydrate contains twice as many hydrogen atoms as carbon or oxygen atoms. All right. Simplest type of carbohydrate with smallest molecules are sugars. All right. Sugars stay sweet. They are soluble in water. One type of sugar is glucose. Right. So glucose general formula is C6H12O6. So 6-carbon compound. Okay. General formula for this is CnH2n. Okay. CnH2n. Okay. So general formula. It's not important. Just remember it for glucose. Okay. So 6 carbon atoms, 12 hydrogen atoms, 6 oxygen atoms. Okay. And atoms are arranged to form a hexagon shape. All right. Now, glucose is the way your carbohydrates are transported around the human body. Obviously, because when we are consuming food, okay. So all of that will be transported to cells, and cells basically utilize glucose, right? And they break it down to release energy. All right. So since it is a simple molecule, it dissolves readily in your blood plasma as well. All right. Now glucose molecules can also link together in chains to form larger molecules. Okay. So in animals, the large molecules that they are formed are called glycogen. Okay. So in animals, it makes up glycogen. This glucose molecules link up together to make glycogen, and in plants they link up to make starch. Basically they're all storage forms. Okay, in animals, even in us, excess glucose is stored in the form of glycogen in your liver. All right. In plants also, you guys must know, it is stored in the form of starch. Glycogen is also called the animal starch. Is this clear? Yes. Everyone else, all good or clear?
Yeah.
Okay, great. Now, plants also make up, another carbohydrate that is called your cellulose. And where is cellulose used in plants? Cell wall. Cell wall. Very good. Plants also make another carbohydrate That is your cellulose. And it is used for making plant cell wall. Okay. Now to figure out that which substance is present in which no which of the substance is present. So for testing their presence, you have a test for starch and a test for sugars, reducing sugars basically. Okay. Firstly, let's talk about a test for starch. So test for starch can be done. So presence of starch can be figured out by using your iodine solution. Okay. In case of positive result, what will the color change be? Does anyone know?
Blue black.
It will change to blue black from
Brown.
yellow brown. Very good. From yellow brown to blue black. Is everyone clear? So we are using iodine solution. Now if I talk about your test for sugars, most sugars is usually for reducing sugars. Now Devasit, where were you?
Yes, I went for auditors.
Oh, okay. Alright, so presence of reducing sugar can be tested by using Benedict solution. And for this test, you need to heat the solution first. I mean, heat the entire solution that contains the piece substance, whatever you are testing for presence of sugar. Okay, first you will heat it, heat the solution, not only the binding solution, the entire solution. Okay, and then heat the solution that contains reducing sugar, and then right, containing reducing sugar, so that it's clear. And then Benedict solution is added. Yes, what is the color change? Anyone knows?
It's from blue to brick red and might come as yellow, green, or orange.
Very good. Excellent. Changes color from blue. Blue to okay. So I'll write the series of colors. Okay, they all show the concentration of reducing sugar present. So it's this the chain goes as blue. 1 second. Give me a sec. Blue to green to yellow. To yellow to orange, red, or brick red. Or brick red. And over here we are using Benedict solution. Is everyone clear with carbohydrates?
Miss, what is the difference between reducing sugar and starch?
Starch is see there are two types of sugars, reducing and non-reducing. Okay, non-reducing sugars give you negative solution, negative results with your Benedict solution. You don't just directly put them. Okay, you basically have to first put them put them in HCl. Starch is basically non-reducing sugar. Okay, it's a separate solution. It's a separate result. Starch cannot reduce other substances. Okay, so there's a separate test for non-reducing sugars. Okay,
Miss,
starch is
aren't both of
non-reducing.
them
Yes.
sugars?
Sorry.
But what's the difference between them? Both of them are sugars.
They are both of them sugars. There's a difference between reducing and non-reducing, right? See, your reducing sugar will always reduce the other substance. Is this clear? Non-reducing sugar will not reduce the other substance.
Okay.
Okay, no, because then this reducing and non-reducing has to do with a lot with the structures that is not part of your scope as of now. Okay, when you will study the structures in A levels, inshallah, of glucose, starch, and all those things, then it will make more sense that why exactly it is reducing and non-reducing. Right? So just remember that the non-reducing sugar cannot reduce other substances. Reducing sugar can reduce other substances. All right.
Miss, and is there a specific like temperature and time that we need to heat the solution?
Temperature, I see that you will do it in A levels, inshallah, when you will practice the practicals. But yes, obviously, you will not heat it at very high temperatures. Just keep them in water bath. Okay. Of about maybe I think 75 to 80 degrees. Okay.
Okay.
And time, obviously, you will keep it until it starts to a bit. It starts to boil a bit at the time when your Benedict solution, when you add it, will give color. Okay. So you will learn this inshallah when perform practicals in A levels. But any other question? Ayla, Ibrahim, Abdul Basit, all clear?
All clear.
Ibrahim, Abdul Basit.
Yes, Miss, clear.
Alright, quickly please note this down, then we will talk about your fats and oils.
Miss, do we have to learn how much percentage of all of this, like the molecules in our body?
Don't learn exactly. It's just to give you a reference of how exactly what is the quantity of each substance. You don't need to learn it. Okay,
Okay.
just for reference. Are you done?
I'm done.
Rest of you?
Done.
Alright, let's move on then. Now let's talk about fats and oils. Alright. Okay, so fats and oils, they are both known as lipids, and fat is basically a lipid at room temperature. Oil is a lipid that is liquid. Fat is a lipid that is solid at room temperature. Oil is a lipid that is liquid at room temperature. Alright, they are known as lipids. Collectively, they are known as lipids. Again, elements, carbon, hydrogen, oxygen, same as that of your carbohydrates. Fat is basically solid at room temperature. And your oils, they are liquid at room temperature. Okay. So they are made up of two kinds of smaller molecules. Okay, one is your glycerol, second is your fatty acids. You have one glycerol and three fatty acids that are attached to it. It looks like this. This is your glycerol. You will have three fatty acids attached to it. This is your glycerol. These are your fatty acid chains. Glycerol, fatty acid chains. And why are lipids and is important? Because they are important for making cell membrane. For making cell membrane. Okay. They basically act as energy stores, or they are used as energy stores, because they form your adipose tissue, that basically is your layer of fat under your skin. So basically they act as your insulation or insulating layer. Okay, so they are insoluble in water, but they dissolve in ethanol. That's important. That's why the test is also your ethanol emulsion test. It's called your ethanol emulsion test. With ethanol to allow any fats in it to dissolve in the ethanol. Then ethanol is poured into a clear tube that contains water. And if the fats in ethanol, they will form tiny droplets in water, which gives a milky appearance. Okay, they form a cloudy milky appearance. In case of presence of fats, in case of positive result, there is a cloudy milky appearance. Okay, firstly you add the food into ethanol and then into water. That's the sequence. And then if it's positive, then it will give you your cloudy milky appearance. Clear? Any questions? Yes. Any questions from fats?
Miss, it will give a cloudy, milky appearance, and also the lipids will be floating on the water.
Yeah, so that is what is giving, cloudy appearance. Those globules, the small fat globules. Yes, you're correct.
Okay, so if the question asks us like what if this like emulsion test was given, and it tells us what tells us that it was a positive result. So do I write that gives cloudy appearance, or do I write the lipids are floating on the water?
Yeah, you can just say a mixture of tiny droplets in the water. They are basically called as your emulsion. And they give you a cloudy milky appearance. Okay. So just say it you will that I will see a cloudy milky appearance, and that is due to the lipid droplets. So if you don't even write lipid droplets, it's okay. The cloudy milky appearance will tell you. Okay, that will be more than enough. That's why the name is your ethanol emulsion test. Is that
Yes.
okay? Great.
Does the body contain oil or just fat?
See, body contains when you're consuming it, you consume it in two forms. Okay, you consume it in fats and oils. We have fat. Okay, it's stored in the form of fat in our body. Okay, that's why we said fat is important for making cell membranes and all. This is usually for fat. Okay, energy stores also fat, insulation layer, everything. Clear, Sad?
Clear, miss.
Okay. Anyone else? Any questions? Ibrahim, Ayla, Abdul Basit, all clear?
All clear.
Ibrahim, you there? All good? All clear?
Yeah. Yes.
Okay, great. Now let's talk about proteins. So proteins contain the four elements: carbon, hydrogen, oxygen, nitrogen. Sometimes sulfur as well, but sulfur is not as common. So you can say sometimes sulfur. Okay. And your proteins are made up of long chains of amino acid. Long chains of amino. Astaghfirullah, sorry. Basically, there are 20 different types of amino acids. And peptide bonds are present between them. They are linked by peptide bonds. Okay, now if I talk about a test, so you will use Biuret reagent test for proteins. It can be done using Biuret reagent. Biuret, not Biuret, please. Okay, in case of positive result, what will it give? It will change color from, yes, What does it change color from?
Blue to purple.
Very good. Changes color from blue to purple. Very good. Purple, or I can also give you violet. Okay, so same shade. All right. Okay. Last thing of the chapter is the structure of DNA. So DNA is the material that makes up genes and chromosomes. Okay, so DNA makes up genes and chromosomes. Okay, and nucleus of every cell in the body contains DNA. All right, DNA contains a code which instructs the cell which amino acids to make at which place, okay, at which point, and which sequence to make proteins. Okay, the sequence of bases in the DNA molecule determines the sequence of the amino acids that are added to build a protein. All right, and DNA is made up of smaller molecules called nucleotides. One second, I'll just reshare this. Just 1 second. Smaller molecules called nucleotides. And each nucleotide contains a base. You have 4 bases: adenine, thymine, cytosine, and guanine. And you have uracil in your RNA. Each nucleotide contains a base. You have adenine, thymine, cytosine, and guanine. A, T, C, and G. So in RNA, instead of thymine, you have uracil. You have uracil instead of thymine. This is for RNA. These bases form bonds according to complementary base pairing rule. And according to that, adenine forms a double bond with thymine. Cytosine forms triple bond with guanine. In RNA, adenine forms double bond with uracil. Cytosine forms triple bond with guanine. Is this clear? Any questions, anyone?
Yes.
Do we need to memorize any diagrams?
Diagrams? Which diagram? This is simple, straightforward. 1 cholesterol, 3 fatty acid. What else? No. You're talking about
We
structure.
need to only memorize this one. Yes, structure.
No. Structure is not important. Okay. Okay, quickly note this down.
Miss, are these bases made of proteins or sugars?
Sorry, which spaces? What are you talking about there?
The bases, are they made up of proteins or sugars?
Hmm. What do you think? What are you saying?
I don't know, probably proteins.
Yes. Okay.
Okay.
Good. Good, Ayla. Nice. Anyone else? Any questions?
What is the difference between RNA and DNA?
DNA is double-stranded. As the name suggests, deoxyribonucleic acid. RNA is ribose nucleic acid. Okay. So RNA is single-stranded. One difference is that, another difference is of your nitrogenous bases. Your DNA contains thymine and your RNA contains uracil. Okay, Sad?
Yes, thank you.
No. All right, done?
Yeah.
Rest of you?
I'm done.
Hello. Abdul Basit,
I'm
Ibrahim.
done.
Abdul Basit? Done? Clear?
Yes.
Ibrahim?
Done.
Okay, great. Great. All right, so we are done with this chapter. Let's start with chapter 5, enzymes. Right, chapter 5. Just give me 1 second. All right. So if I ask you to define enzymes, how would you guys define enzymes? Yes, one by one. Yes, Zoya, go ahead.
Enzymes are biological catalysts that are not used in the reaction. It speeds up the processes of either breaking down a reactant or building it up.
Very good. So how would you describe enzymes?
Proteins which act as biological catalysts, speeding chemical reactions inside the body.
Very good. Ayla, what about you?
They are biological catalysts that play a role in metabolic reactions.
Very good. Okay, Ibrahim, go ahead. Ibrahim,
Yes.
how would you define enzymes?
Enzymes are proteins that act as biological catalysts by
Right.
speeding up chemical reaction.
Okay, Abdul Basit, what about you?
Enzymes are biological catalysts and they are proteins which speed up the rate of reaction.
Very good. So all of you said some similar thing, biological catalyst speeding up the rate of reaction without being used up in the reaction. One thing that I would like to add is that how do they speed up the rate of reaction? That is by lowering the activation energy required. Is this clear? Lowering the activation energy without being used up in the reaction. Is this clear? At this point, just add this in your definitions by lowering the activation energy. How are they speeding up the rate of reaction? By lowering the activation energy. Is this clear?
Yes.
Very good. So for example, you have hydrogen peroxide. I hope everyone has heard about it, H2O2. So there's an enzyme called catalase that breaks it down into water and oxygen. It's a very famous enzyme, okay? Breaks it into water and oxygen. Enzyme is catalase. Okay, H2O2. Go down into oxygen, water and oxygen. Just balance it. Balanced. Right? Now if I talk about naming of enzymes, all enzymes, majority of the enzymes end in ASE. Agreed? Agreed, yes or no?
Yes.
Right? For example, enzymes that catalyze the breakdown of carbohydrates are called carbohydrates. And then that catalyze the breakdown of proteins are called proteases. Those that catalyze the breakdown of lipids are called lipases. Agreed? Those that, now if I talk about further about carbohydrates, you have amylase that break down that catalyze the breakdown of starch. You have maltase that catalyze the breakdown of maltose. You have sucrase that catalyzes the breakdown of sucrose. Is this clear? Sucrase, sorry. So can you see
Clear.
all of them are ending in ASE? But there are some that do not end in ASE as well. An example, can you think of? Any enzyme that does not end
Pepsin?
in AS. Very good, pepsin. Anything else? Similar to pepsin.
Amylase.
I'm asking it does not end in ASE.
I think one of them is called trypsin or something like that. I forgot.
Trypsin, very good, yes. Similar to pepsin. Pepsin and trypsin, okay? These are on your syllabus as well. Right, so naming is clear. Majority end in ASE, but there are some that do not end in ASE, okay? Do not follow general trend. Okay. Now, let's talk about the factors that affect your enzymatic activity. Or factors that affect enzymes. Focus on two factors. Enzymes are very sensitive to temperature and pH. Yes, if I talk about temperature, so I will say as temperature increases, rate of enzyme activity also increases. General statement. Rate of enzymatic activity also increases. And why is that? As at high temperatures, okay, you can break it down into bullets as well. I'm writing it into one paragraph form. Substrate molecules. What are substrate molecules? Substrate molecules are those molecules on which your enzymes are acting upon. Is this clear? Substrate molecules gain kinetic energy and they move faster and collide with one another and have more, I can say, have more and more successful collisions and have more successful collisions with substrate, with enzymes. Okay. This results in formation of more and more enzyme substrate complex. And therefore more product. Maximum activity is achieved at optimum temperature. However, above optimum temperature, enzyme starts to denature and loses their shape of active site. Therefore, product formation stops. Above optimum temperature, enzymes start to denature and lose their shape. So, your enzyme and substrate stay no longer complementary because, they lose their shape. So, product formation stops. Product formation stops. Is this clear?
Yeah.
Let me just copy a graph and show you how it looks like. Let's show you how it looks like. It's here. Let's go to chemistry. Alhamdulillah. So, temperature increases, rate of reaction increases, optimum 37, and then it starts to drop, and then your enzyme denatures. Now, if we talk about pH, All right. So in pH, so I will similarly start same way as pH increases, enzymatic activity also increases. Okay, maximum activity is shown in the optimum pH. Okay. Now in this case, you have no kinetic energy or something like that. Okay. So you will say HRDs in bullets, enzymes show below optimum pH. Maximum activity is seen at optimum pH. So it differs with different enzymes. Okay. Below optimum pH, enzymes become inactive. Denatured. Look at the word choice. I'm saying they become inactive. It means that if you increase the pH, they will return to normal. You cannot return a denatured enzyme to normal. You can return an inactive enzyme to normal. But above optimum pH, enzymes become denatured. And lose their shape of active site. And since again your enzyme substrate are no longer complementary, product formation stops. Just like here. So product formation stops. And look at these graphs. This is a graph whose optimum pH is 7. Okay, look at the activity. Initially it's inactive. Inactivity, it's okay. Okay, you can increase the pH and the enzyme will work again. But if it becomes denatured, okay, if it loses its active site, you guys know, right? Enzymes and substrate work according to your complementary, lock and key mechanism. I'll just talk about working of the enzyme as well. Okay. So then they will no longer stay complementary, so they will no longer fit. So no longer product conformation will occur. Now look at this graph. In this, look at we have pepsin. Pepsin has an optimum pH of 2. Right? That's why we have no optimum pH. In temperature, we have an optimum temperature approximately 37 degrees Celsius. Okay, but it differs. Last thing is the working of enzyme. So enzymes work according to lock and key mechanism. Enzyme is your lock. Let's say this is your enzyme. Let's say this is a substrate. For example, this is your active site. Okay. It will fit here. It forms enzyme substrate complex. And then let's say it broke it down further, and then you have your enzyme and product separate now. Let's say they're two small units now. For example, okay, this is your product. Enzyme is back to normal. Again, ready to catalyze another reaction. Okay, so enzyme and substrate need to be complementary. Enzyme and substrate are complementary to one another. Like for example, this will not fit here, this will not fit here, okay, because they're not complementary to the shape of the enzyme. Like you have a specific lock, a specific key for a specific lock, right? Not every key fits into every lock. Similar to that. Is this clear? Any questions, anyone?
Yes.
All right, any questions? Shad, Zoya, Abdubasit, Ibrahim, all good, all clear?
Yes.
Clear, miss.
Abdubasit, Ibrahim.
Clearness.
All right, that's great then. Okay, please note this down. Should I move down? Are you guys done?
Yes.
Rest of you?
Yes.
All right. Okay, okay. All right, please quickly note this down. All right, guys, done?
Just give me 1 more minute.
Sure, sure. Okay.
Miss, for the pH diagram,
Yes.
the red one written pepsin, and what's above it?
Above it's protease. Okay, the class is protease. And the specific enzyme is pepsin. Okay. Protease is for all peptides. All right. Pepsin, trypsin, peptidase, all of them come under protease. Okay, we are going to talk about this in digestion chapter, inshallah. All right, so clear?
Yes, thanks.
No problem. Anyone else? Any questions? I love the Basil, Ibrahim, Saad.
No.
No.
All right, great. So today we completed of 2 chapters, biological molecules and enzymes. Tomorrow, inshallah, we are going to start off with chapter 6, and I think that will take approximately 1.5 class. So we're going to be starting off with a bit of large chapters from tomorrow, inshallah. First, we'll do plant nutrition. Yes, chapter 6 is plant nutrition, so we're going to start off from that, inshallah, tomorrow. Are you guys done?
Yes, miss.
All right, then. So that's all
Yes.
for today. Okay, great. So that's all for today. Then I will see you guys tomorrow, inshallah. We're going to start from chapter 6. See you then.
Allah Hafiz.
Allah Hafiz.
Allah Hafiz.
Allah Hafiz.